Hello!
Dissection of Drosophila larvae and immunohistochemical analysis of imaginal discs.
First, we disinfect the surface with alcohol. In the first while, where the cross between two different Drosophila lines was performed, larvae have started to appear. We transfer the adult individuals, that is, the flying flies, to another tube so that only larvae remain in the first one. We collect ice using the generator. This is necessary to cool the larvae, making them less mobile and more manageable. The first step is the collection of eye discs. We add 100 microliters of PBS into each well and transfer the larvae into the filled wells. Don't forget to take notes to remember which genotype is in which well. As you can see, in some genotypes the tappy balancer is present. The plump larvae expressing this balancer should be discarded. We drop a little PBS onto the glass slide and mix the larva in it. Using the first pair of forceps, we grab the larva's head and with the second one the body and pull! To be more precise, we are interested in the antennal eye imaginal disc because this structure contains the precursors of both of the eye and the antenna. This is how the dissected part of interest looks. Next, we need to dispose of the PBS. This must be done carefully. Fixation is done using 3.7% formaldehyde solution. In a microtube, we mix 244.5 microliters of PBS with 55.5 microliters of formaldehyde. We then add 100 microliters of the resultant mix into each well. This step is necessary to fix our samples. We move the samples into a box and leave them to incubate for 20 minutes. To save time, we can now prepare calculations for the primary antibody mix. The incubation time is over, so we return and retrieve our box. Formaldehyde is toxic, so for its disposal, we use a special container. All samples need to be thoroughly washed. We do this by editing 100 microliters of PBT into each well, 3 times. Since I'm not alone in the lab, we label our box. The first wash is complete. Since the solution still contains formaldehyde residues, we dispose of it in the designated waste container. For the second wash, we again add 100 microliters of PBT into each well and incubate. Each wash takes 10 minutes. Time's up! The used solution can now be poured into the general liquid waste container. We perform the third wash, 100 microliters of PBT and incubation. During this time, I prepare the primary antibody mix, since the next step will be their application. I retrieve HRP and Repo from the fridge and Caspase 3 from the freezer. While I'm mixing everything, I will explain what each of them is used for. HRP labels axons, Caspase 3 marks apoptosis, and Repo marks glia. I am studying changes in the size of the Drosophila eye under different expression levels of the dead protein. In this experiment, I use flies obtained by crossing with the genotype optic skull 4 US dead GFP. As you can see, I've already added the primary antibodies and now I'm leaving them to incubate overnight at 4 degrees. Second day, we go to the cold room and retrieve our box. We will wash out wells 3 more times, again using PBT. The washing protocol is the same, nothing changes. So I have time to explain the experiment in a bit more detail. Biological processes are inherently variable, but the outcomes of development, such as species-specific organ size and shape, are usually remarkably precise. This precision suggests the existence of feedback mechanisms that minimize deviation from a target size. However, we have very limited understanding of how this mechanism works. In this study, we focus on the question of organ size precision, using the Drosophila eye as a model. The size of the adult eye depends on the growth and differentiation rates of retinal progenitor cells, which are in turn regulated by proliferation and apoptosis. Specifically, apoptosis of progenitor cells is suppressed by DPP, a ligand of the TGF-beta signaling pathway, which is transiently produced by early differentiating retinal cells. The dead protein is an inhibitor of this DPP signaling pathway through inhibition of a key element, MET protein. Returning to our experiments, we need to consider and insert the secondary antibodies. These are rabbit Alexa 568 for caspase-3 visualization and mouse Alexa 647 for repo. HRP already contains gold secondary antibodies, so we don't need to worry about. Let's prepare a mix for secondary antibodies. So, returning to the experiment, the size of the adult Drosophila eye depends on the growth and differentiation rates of progenitor cells in the eye imaginal disc. We know that the growth rate of progenitors is a result of the balance between their proliferation and apoptosis, with the latter contributing to both the final size and shape of the eye. TPP prevents apoptosis. Therefore, when we increase the expression of that, a DPP inhibitor, DPP signaling decrease, apoptosis and the apoptotic index increase and the eye size decrease. Which exact apoptotic mechanisms are triggered by these changes is still unknown. This question requires further investigation. But for now, we aim to track at least the correlation between the apoptotic index and changes in eye size. Regarding the video, I've already performed the secondary antibody incubation. For that, I had to wait for an hour and a half. Now I'm finishing the first wash, which needs to be done after the secondary antibodies. Everything is the same, PBT solution, 100 microliters in each well. It's important that all the samples we're interested in are fully submerged in the solution. Also, if bubbles appears on the surface, we can pop them with forceps. We pour out the old PBT and add new one. It's over, so let's continue with the third wash. Meanwhile, we can prepare for the final step. It's fixation. With this, our samples can long for more time. We take the Vector shield out of the fridge. 10 minutes of the last wash are up, so we go and get our box. Now we dispose all the PBT we have. We need to make the wells as dry as possible. Vector shield is used for mounting the samples. We used to use glycerol, but Vector shield turned it out to be better. The next step will be done the following day, so we put the box and the Vector shield into the fridge and wait till tomorrow. Hello! Third day! Glass, 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 glass. Pencil? We take the glass slides and label them. 3 times, 3 genotypes. We add a bit of Vector shield to the first glass slide. On the second, the working slide, we also add Vector shield and transfer our samples to the working surface. At this stage, we need to remove all unnecessary structures and leave only the eye imaginal discs. This is what they look like after processing. We transfer them to the corresponding slide and cover with a cover slip. We repeat the same steps for the second genotype. Again, we add Vector shield to the slide we're interested in, as well as the working one. We transfer the samples. This is how they look. We search for the eye antennal discs that need to be cleaned. Here they are. We transfer everything to the glass slide and cover with a cover slip. And for the third control group, we do the same. We isolate the discs, transfer them to the appropriate slide and cover with a cover slip. This process is actually very time consuming because it's quite difficult to pick up the discs with forceps. Especially when the forceps have chipping tips. And I haven't even mentioned how fragile the structures are. To make sure the cover slip doesn't move and accidentally damage our samples, we use nail polish. Tada! We are reaching to the confocal microscope. Here I'm showing the slide. We need to flip it because the objective is located underneath. We secure our glass with clamps. Lower everything down, close the lid. So now we can begin. We turn off the lights and turn on the DAPI filter to find our disc, or at least some structure. We move the slide like this. And here we'll lower and raise the objective. We adjust the focus and find the first disc. This is what it looks like. just like in the picture. Here is the program we are using, LS from Leica. We press start and wait for the image from each laser to load. The working panel. There are many different parameters you can adjust, and depending on that, the image will change. It's essential for me to align the image properly because I will be using it later in the figures for my work. Overall, this is what the images look like. In green, we see RIP antibodies, glia. In red, caspase-3, apoptosis. And in the blue, HRP, which are axons. Of course, in the program we can change the laser spectra, the display colors and their intensity. Working with the confocal microscope takes quite a bit of time. One slide takes me between 30 minutes and an hour, depending on the angles I need for the photos and how many discs I managed to isolate. As you can see, the experiment itself is quite time-consuming when you consider all the stages. Here I take a tissue and wipe off the water. I used a 40 times objective. I lower the objective, close everything, check that everything is okay. Turn off the laser and that's it. The work is done. Oops! Thank you for watching.